Hello, we're going to continue with Chapter 4, which has the broad topic of protein structure and function. We're going to look now specifically at how proteins are controlled. And we can look at different aspects of control, like their activity, their location, what they bind. All of these things are going to be regulated by the cell. One example we'll use is enzymes. We're going to see that enzymes are efficient. Many enzymes are made within the cell, but are kept inactive. So you go through the process of transcription, translation, folding, and the proteins are there, the enzymes are there, ready to be activated. But they will only be activated when needed. So we're going to look at how this one example of regulation in a biosynthetic pathway. So in a biosynthetic pathway, we have multiple chemicals that are initially a substrate, but then can become a product, and then a product can become a substrate again. So what do I mean by that? So here A is a substrate, right, starting material that is needed to make B. B was the product of the first enzymatic reaction, is needed to make C. It's a substrate used by enzymes to make either C or X. The arrows in this pathway represent enzymatic activity. So there are enzymes working at each of these arrow points. So this is a hypothetical biosynthetic pathway where there are two branches. So again, we would start with substrate A, which can be converted to B by something I'm calling enzyme 1. So right, B then could be converted to X by enzyme 2 maybe. And in this situation, we're going to assume that we only have a limited amount of B. But we're also going to assume that enzyme 2, and we'll call this enzyme 3, have the same affinity or the same likelihood of binding to the substrate B. And so that under normal circumstances, the amount of C and X produces about the same. However, we're going to look now at a situation where one of the metabolites affects its own production. So here we have the metabolite Z. A is converted to B, B to X, X, there's an enzyme that converts it to Y, and Y, there's an enzyme that converts it to Z. Z then goes back and has an effect on the enzyme that converts B to X. As you read this diagram, you can see that Z, there's a line that coming from Z with a bar at the end. This bar is a common symbol for inhibition. So when you see a line like this, the line ending in a bar, that suggests inhibition or a negative effect on, for example, enzyme activity. In contrast, if this line ended in an arrow, that would suggest positive regulation and activation. So what we have here is Z inhibits the activity of the enzyme that converts B to X. As we have more Z present, we're going to shut down more and more of the enzymes that convert B to X. As levels of Z increase, we're going to see a decrease in the production of X, a decrease in the production of Y, and a decrease in the production of Z. So what would be the kind of answering this question here, what would we see as an effect on X? Well, we should see a decrease if we inhibit this enzyme, stop it from working as well, have fewer of that enzyme that's active. So if we normally have 1,000 enzymes in the cell and maybe 900 of them are active, as we increase levels of Z, fewer and fewer of those enzymes would be active, which would result in less production of X. If, as we said before, B was in limited supply, and now we're no longer making X, we would expect that levels of C would actually increase because this enzyme, as long as there's plenty of it around, now has more substrate to make C. In this image here, we have a biosynthetic pathway for production of amino acids in bacteria. You may recognize some of these amino acids. Methionine might be the one you might pick up most, right? Our start codon, isoleucine, threonine, lysine. You can see from this image, each of these amino acids is made from precursors, chemicals in a biosynthetic pathway. So aspartate is converted to aspartate phosphate, converted to aspartate and semialdehyde to homoserine, which can then be converted to threonine, and threonine through a series of enzymatic reactions can be converted to isoleucine. So here we have something similar to the hypothetical where we have one chemical in the pathway that can be a product of the first enzymatic reaction if we're looking at aspartyl phosphate, but also a substrate for the next one. And again, different products, threonine, for example, is something that's active in our body, but is also a substrate for making other active metabolites in our body, isoleucine, for example. I have this area circled because we're going to focus in on that to look a little more closely how negative feedback can work. We see many arrows here. Aspartate has an enzyme that converts to aspartate phosphate, aspartate semialdehyde, and homoserine, and then there's that negative feedback loop, and we're going to focus in on that. So I've drawn this pathway out just looking at these few steps, given each of the names of the metabolites and tried to draw them as different shapes so we can kind of keep track of what their shape might be in relationship to the enzymes that are involved in producing them. So in the second part of the drawing, I have aspartate. I've tried to draw a representation of the enzyme that might convert aspartate to aspartate phosphate. And you can see that this is the active site of the enzyme binds to the shape of aspartate. So aspartate will bind to the active site, and this active form of the enzyme will convert aspartate to aspartate phosphate. Aspartate phosphate can then bind to the next enzyme in the sequence. You can see that the active site for this enzyme matches the shape of aspartate phosphate. Aspartate phosphate will be converted by this enzyme into aspartate semialdehyde. I haven't drawn the third enzyme in the pathway, but it would be working right here and convert aspartate semialdehyde to homoserine. Homoserine is going to be the metabolite that causes negative feedback to happen, or feedback inhibition. So homoserine, it turns out, can then go back and act upon the enzyme that binds aspartate as its substrate. When homoserine binds to the enzyme at this spot right here, which is an allosteric site, so it's a site that's different than the active site. So here's the active site right here. This is the allosteric site. Homoserine binding to the allosteric site causes a conformational change in the enzyme. The conformational change closes off the active site of the enzyme, and the enzyme, when it's in this inactive form, cannot bind aspartate, and it cannot produce aspartate phosphate. As levels of aspartate phosphate drop, so would levels of aspartate semialdehyde, and so would levels of homoserine, right? Which would mean as there's less and less homoserine, more of the enzyme would be in its active form. So this is an example of the mechanism. So there's a key word there. Mechanism meaning kind of the steps to make it happen. The steps to make this inhibition happen is that the homoserine binds to the allosteric site. This causes a conformational change that results in an active site that can no longer bind substrate. So we're looking at allosteric regulation of enzyme activity. In our example, the product homoserine is going to bind to the first enzyme in this pathway at the allosteric site. Binding at this allosteric site causes a conformational change in shape, and when the enzyme undergoes this conformational change, the active site, right, which was the site here, is no longer available to bind the substrate aspartate, which makes it an inactive form. If there's no longer any homoserine available, we would reverse the conformational change and the enzyme would once again be active. In the textbook, they give the example of aspartate transcarbamylase as an enzyme that is controlled or regulated via an inhibitory feedback. So in this case, the picture here, your left is the active enzyme. You can see it says on, and you can see they've highlighted the active site. When CTP is present and binds to the enzyme, you can see there's actually 4 binding sites. The active site is closed off. So again, this is allosteric regulation because we have a molecule binding at a site other than the active site, and it's causing a conformational change. And in this case, the conformational change is affecting the active site so that it can no longer bind to substrate. So this would be inhibition. So summarizing this, cyclic AMP binding site in the previous slide is an example of binding at an allosteric site. It's binding at a site other than the active site. This causes a conformational change which regulates the activity of the enzyme. The example I showed you in my drawings and the example in the previous slide are both examples where allosteric binding inactivates the enzyme, but there are also examples where binding an allosteric site actually flips the enzyme on or activates the enzyme. So we're going to look at an example of that next. So here in green is the enzyme. This is an enzyme that oxidizes sugar as a step in cellular respiration. And its allosteric regulator is ADP. ADP, adenosine diphosphate, will build up if cellular respiration is low. So you have a buildup. We need to convert ADP right into ATP. ADP will bind to the enzyme and cause a conformational change. So this is one conformation and this is the other. In the absence of ADP, the enzyme is in an open and inactive shape. When ADP is present, the enzyme changes shape into a way that it is likely to bind the sugar, which is blue. So we're getting a switch from an inactive to an active form in the presence of the allosteric regulator. So this is kind of repeating that. Enzyme is in inactive to start. ADP binding switches the enzyme to a closed or active position. The enzyme can bind the substrate in this closed position. And the nice thing about this image here is that it's showing you that there isn't just one enzyme in your cell, right? There isn't just one of these enzymes working, there are many. And in this picture, they're showing you 10, and they're saying in the absence of ADP, very few or 10% of the enzymes are in the active conformation and able to bind sugar. As cellular respiration rates drop and ADP builds up, we now see ADP bound to 100% of the enzymes, and 100% of the enzymes are active, breaking down or oxidizing that sugar. So again, this is allosteric regulation, but the difference is that the allosteric regulator is activating the enzyme rather than inactivating the enzyme. What's in common? Binding at an allosteric site, causing a conformational change, and that conformational change is affecting the active site.